Sufficient rooting densities are crucial for accessing water and nutrient resources and improving crop resilience in a changing climate.
By measuring crop roots at different soil depths, we can see how successfully the plants have explored the soil environment, and this helps us to understand how roots are influenced by factors such as cultivation, nutrition, weather conditions and genotype.
This video demonstrates how to collect soil cores from field trials to measure crop rooting down the profile. But these samples can also be used for different purposes, such as measuring soil characteristics or soil nutrient contents at different depths.
To collect the soil cores, a mechanical soil sampler is used, which can be self-propelled or towed on the back of a vehicle. The sampler uses a pneumatic hammer to push the auger into the soil and lifts it out of the ground with hydraulics. A steel auger is attached to the sampler to collect the soil core. In this example, a metre-long window auger with a diameter of 26 mm is being used. Before the soil core is collected, make sure you have the appropriate safety equipment for the job. As a minimum, this should be steel-toed boots, ear defenders and gloves. For collecting root cores, it is recommended that samples are taken when the root system is at its maximum size. In this example, samples are being collected from a cereal crop at early grain filling. Collect samples from one end of the plot to reduce the amount of damage caused, particularly if later measurements are to be collected, such as final yield. The diameter of the auger being used determines the number of samples needed to ensure it is representative of the crop. For augers with a diameter below 30 mm, it's recommended to collect 6 samples per plot, and for augers wider than 30 mm, 4 cores per plot can be taken. The samples should be split between samples taken both within and between rows for row crops. To measure crop rooting down the soil profile, it's necessary to collect soil at different depths. In this example, soil is collected in 20 cm sections, so 5 individual samples are collected per metre deep soil core. The soil is placed in labelled bags and stored in a cool box, which is taken back to the lab, where the samples are placed in a freezer to prevent rapid degradation. To wash the roots from the soil, please refer to our tutorial video, which provides details on how to extract and analyse the roots from these soil cores.